regular attenders know that FISH is our Friday informal seminar hour. So today's a little bit more formal because we are lucky to have two speakers join us today. And we're celebrating a groundbreaking discovery here at Bridgewater, the discovery of Hormesis. So 50 years ago, in plant phys and in research, Dr. Ed Calabrese discovered this concept. So we're very excited to welcome his colleague, Dr. Walter Kazumbo, and also Dr. Calabrese home, fitting that it's homecoming weekend, to Bridgewater. And I also would like to extend a very warm welcome to the members of Ken Howe's family who've joined us for dinner last night. We have joining him his Ken Howe's wife of over 50 years, Pat. We also have his son Spencer Howe and also his daughter Rhonda. So we're very happy to have you guys here with us at Bridgewater. So our first speaker is going to be Dr. Walter Kazumbo. He's a close colleague of Dr. Ed Calabrese, who is a witness to his beginnings of the field of Hormesis and its role in mandating regulatory standards. Dr. Kazumbo, he earned his bachelor's degree in biology at Princeton University, and then he went on to earn his master's in science and biology at Purdue University. He then earned both his master's in liberal arts and doctorate at Johns Hopkins University, and went on to a postdoc at the Swiss Cancer Research Institute, and then to the Center of Environmental Medicine and Lung Biology at the University of North Carolina at Chapel Hill. Ultimately, his passion for toxicology led him to work for the Environmental Protection Agency in Washington, D.C., and to work for over 20 years as the program manager for Math, Information, and Life Sciences at the U.S. Air Force Office of Scientific Research. And he retired in 2011.
Yeah, that's good enough.
Okay, so thank you very much.
Welcome everybody, and I want to thank Dr. Kowalski for inviting me and giving me the opportunity to speak about a close friend and a great scientist, Ed Calabrese. I feel honored. I hope I do him justice. Okay, so she already told you about me, and I want to talk about Ed relative to Hormesis and the science program that he's doing. So I really had three topics. I wanted to get Ed, a story about Ed, about his program, the science that he's done, and a little bit about the rudiments of the science too, just to understand. Because I hear Ed is going to be very personal in his approach, and so we'll get a little bit of science in this too. Now, at the end, if I run a little bit long, I can dump some of the slides at the end that are future directions, but I think you'll get the meaning of it. So, the overview. Well, we'll talk a little bit about why the Air Force is interested in this, and how I got to meet Ed. We're going to talk about some of the dose response models that you have to understand Hormesis. Talk about Ed Calabrese's program, his accomplishments, etc. Give you some great examples that Ed got from his database on ionizing radiation, Hormesis, and then also chemical Hormesis. A key finding that Ed he sort of unified the field of Hormesis by showing the adaptive response, preconditioning response used in the medical areas. They're all the same as Hormesis. People just didn't understand it and talking different language, the language of tongues, and nobody's understanding it. Then with a new Hormesis research directions that I think are going to be exciting. We really haven't gotten too far into that, but it has to do with not ionizing radiation, but non-ionizing radiation. And something that we think will have a lot of medical, therapeutic, protective, and enhancement effects that have not been investigated up to this point. And then some summary comments. All right, so back in the early 90s, I first got my job in the Air Force, and I was given a $13 million program per year to set up on environmental quality. A lot had to do with environments that the Air Force had, and they were exposed to this, ionizing, non-ionizing radiation, chemicals of different sort, and they wanted to set some standards. Well, then you have to go, okay, well, how do you set a standard? It's based on what's toxic and what's not toxic. And if you go back and look at the dose response models of the time, basically they do all their studies at high doses and extrapolate down through zero. And that basically is an extrapolation, not an interpolation. So they're just assuming that when you get close to zero dose or around zero dose, you're going to get some kind of effect. So, but that's all, they don't know exactly what the effect is at the low dose because nobody does the studies. So they take guesses, they put safety factors in there, 100, 1,000, And they're assuming that they're going to meet. And it's very conservative, so they probably will meet that. At the time, I was aware of the term hormesis, had no idea what it was about. When I found out it talked about relative low doses and some kind of stimulus response, I said, gee, I should find out more about this. At that time, I was aware that Ed was putting together an NIE, National Environmental Health Institute conference down in North Carolina. And I said, gee, I ought to go there and just check it out and see what this is all about, because I'm interested in low doses if we're going to have some meaning to this. And I go down there and I see these curves, and I'm going, what? This can't be right. I mean, you're telling me you get way down in the low dose area and you're going to get a stimulation instead of no response or some kind of toxic response? Doesn't make sense. I said, I got to go down and see what's happening down there. Okay, so I went down there and I saw the curves, and I talked with Ed, and I said, gee, you know, is this stuff true, Ed? And Ed says, well, you know, I'm really interested in this, and I don't know if it's true. I want to investigate whether it's true or not true. I think this is an important thing. And I said, you know, I sort of agree with you. And I said, when's your next conference? And he says, I don't know when the next conference. It may be 2 years, 3 years, 4 years. I said, what? I said, this is extremely important. Why is it taking so long? And he says, well, I've got, you know what it is to be a research professor, right? You got classes, you got students to advise, you got meetings, and he's got grants to ride up the hill to keep himself going. And he says, I got to go out and find funding for this, you know, 5,000 here and 5,000 there. And I said, okay, this is probably a pretty good thing for the Air Force to get involved in, because if we can make some progress in this, we're going to get some answers. So I said, why don't I just fund you on a yearly basis for 3 years anyway, and see where it goes. And you send me in a proposal, etc. And what are the research questions? What are the importance of these questions? It's fundamental, basic research questions. This is in high toxicology, high science. We're looking 10, 20 years out. So, that's, he says, fine. So we started there, and our relationship lasted longer than 3 years. So here I am in his honor. So I'll show you a little bit about, first of all, a background on, you know, it's an alternative model, a dose-response model. Okay, so we have basically today, we have two dose response. One is the linear one. That's for carcinogens. So any chemical carcinogen or any ionizing radiation that potentially could cause cancer, we'll use this. So you notice that the only safe level is at a 0 dose. And then the second model they use is for non-carcinogens. Anything, and there you have a threshold response. And this line here is the control level or the 0 equivalent point. And basically, you know, nothing goes below, if you go below this line, you're actually going to, if that's a toxic response going upward, if you go below this line, you're actually going to have a healthier response. So nothing goes. So we have this zero-based linear, no threshold model, and then we have the threshold model. Hormesis now comes out with the idea that you have a low-dose stimulation and a high-dose inhibition. And there are two types of hormetic curves depending on what the response is. If it's a growth type of response, such as looking at how tall the plant's going to grow, or if it's how many cells are going to grow in a petri dish, you're going to see the beta curve. This curve right here. And it shows that if you start at some very low 0 dose, you just get control level right there. And as you move the dose up, you don't get a response until you hit some low dose that is stimulatory. And then if you get out to this point, it becomes toxic, and then you're in an inhibitory zone. Now, if you're looking at a disease like cancer, then you're using a disease-related endpoint. It's called the J-shaped curve. So the tumorigenesis is upward, let's say, and if you go below the controls, that means you're better off. That means you have fewer spontaneous tumors than the control, which is exposed to 0 carcinogens. And here, once again, if you have a dose of a substance, it says that, let's say it's dioxin, And if you're down very low, you're not going to see anything, and you get to a stimulatory dose, which actually cause fewer tumors. If this were a tumor, the tumors would go below the control level. And then it would move up as you increase the dose so that you then get in the carcinogenic area. So that's hormesis, beta curve and the J-shaped curve. So if we overlay the hormetic curve upon the linear no threshold model and the threshold model, we see that we do come down below the control level, indicating we have a hormetic zone. And between the hormetic zone and zero dose, there is a dose that will not do anything, really won't modify the response of the organism. In this case, this is J-shaped, so we're looking at, let's again say again, tumors growing, the number of tumors. But then here it's saying that, so you're telling me that if you give something like low-dose ionizing radiation at a low-dose level, we're going to see fewer tumors in the animal instead of the same level as control? Say control had 10, we're going to see 5. That's saying it's doing something that doesn't make sense, really. It's supposed to be a carcinogen. And then the hormetic dose range is how far across that dose, what the dose is that actually gets you into the hormetic response. Once you get beyond that, though, you get into no observable adverse effect level. That's the threshold, more or less, for the hormetic curve. Then you get into a linear phase. So as I was saying before, the EPA and other agencies right now, when they test animals, they take points up here, way on the high end. And they assume if it's a carcinogen, they say, okay, we got a point here, we got a point there, we got a point there. Now, we'll draw a line through here, but this straight line has to go through that point and through the 0 point. That's what, so they force it through. That's an extrapolation. So they're extrapolating from high dose to low dose, assuming they're at the low dose, it's all linear. Ed's point was, and has been, and he's been working for so hard and so long, is to show us, hey, it's a little different. It's not that simple. So what is his program? Got involved with Ed, I said, in 1998. It's 2016, so I thought I'd first hit some of the major metrics, program metrics we call this. So what did he do? What's his output? He's written over 170 publications on hormesis. That's not his total numbers. It's probably up around 400 or 500, but 170. So he's putting them out each year. He's invited he's been invited to give presentations at 250 or plus, and that's not the number of invitations he's turned down. So, you know, if he were 2 people, if we were twins, you'd probably see 500 there instead of 200. He's been interviewed by radio, newspapers, TV, YouTube, etc. He's been, and this is especially after around 2004 and 2005, he made a breakthrough. And he showed that that chemicals actually, that the hormesis should be the default dose-response model, not the other 2 models. And so Science, Nature, etc. came and interviewed him. He's held conferences, international, 14 since then. You bring in speakers outside, 2-day conferences, 3 hormesis workshops. At the beginning, when he started this off, this is typical Ed. He says, you know, I'm really skeptical about this whole thing. We're here, so I'm taking the point that it may not be true. So he brings in the critics of hormesis, and we have a 2-day meeting on a workshop. And they sit there and they pummel him as he presents all the information for 2 days straight. And he goes, and they go away. And the idea is they had to write a critique of it and send it back to him. So they send it back to him, and he looks at the critique and says, oh, this is all the things that they think are fine. And then so he says, this is what I'm going to address. I'm going to address these points that the critics have. So if I can address the critics' points, that should help validate hormesis or invalidate it if it's not true. So that was the approach. So these workshops were extremely valuable. Collaborators, right now there's 11 national, 8 international collaborators. These are collaborators that he's published with mostly. Europe is really interested in this and constantly asks Ed to come over and speak. He's the founder and editor of Dose Response Journal, International Dose Response Society founded. He's got an award since 2009, Marie Curie Prize in Radiation, the award for cell-cell communication from England, honorable doctorate from McMaster's University, and this is a new one, Ed, you're going to have to tell me what this is about later, Beckman Award for Doctors of Disaster Preparedness. Anyway, you can see he's done a lot in terms of the metrics. Okay, what are the major questions, Ed? What would we have? These are only some of them, but he says, well, is hormesis real? Is it a real phenomenon? We got to find that out. How generalizable is it? Does it apply to certain endpoints, certain species, certain types of chemicals, certain types of radiate? What's the deal there? What percent of chemicals, if it is hormetic, are actually hormetic? And that is it 1 out of 1,000? Is it 50%? Is it 99%? If we could do the studies. What are the quantitative features of the hormetic dose response? How broad is it? How high can the amplitude go? What are the mechanisms of action? As scientists, we need to understand how this works, at the biochemistry level, the genetic level. Let's see, what are the scientific origins of the linear no threshold model, okay? Because where did this come from? This going through, where did they decide that it just goes through zero? What's the evidence in the data that shows that at low doses you actually have a straight linear line? So he's gotten to that, into the history, and that takes him into Nobel laureate investigation, Herman Muller, and he's still writing on that, and he's published a number of great articles on this. And so that, because if it wasn't validated, that gives credence to the hormetic model if we can validate the hormetic model, which Ed has been doing for 20 years or more. Okay, so is hormesis related to other responses, like the adaptive response conditioning, U-shaped curve? There's a whole ton of these things all over the place, and they all think they're different depending on, if you're in the medical field, you're looking at preconditioning. If you're a normal biologist, you're talking about the adaptive response. If you bring up hormesis, it scares people away. So they all think different things are different. So he's going to say, what is the relationship? Now, what is the relationship to epigenetics, this new field? Because this new field doesn't talk about gene mutation, it talks about actually changing the expressions of the genes you have. And that is an environmental, it's huge environmental impact upon it. And what are we talking about? We're talking about the effect of the environment, whether it's ionizing radiation, chemicals, pressure, heat, you name it, electricity. And then the new area we're going to talk about, because we want to move into looking at non-ionizing radiation, I mean light, electromagnetic fields, radio frequency radiation, microwaves, etc. This is an area the Air Force is interested in. And we're not looking at it, we're looking at it, remember, if it's hormetic, we want to take advantage of that hormetic zone because that could be beneficial. Not all the time. I mean, because you could actually stimulate something and the stimulation you're doing is bad, if it's an endocrine function, for example. Or if it's antibiotic, if it's a bacteria and you're stimulating a bacteria, will the bacteria repopulate your body at a faster rate? That's bad. So it's not necessarily good, but in many cases it's good. You have a therapeutic pathway. You should understand what the biological molecular events going on in this hormetic response zone are. And that takes us to the translational biology and medicine, etc. If we can understand that, we could actually use this as a translational science study. Now, the approaches he took, he brought in workshops. I already talked about that. Annual international conferences, I talked about that. Collaborate with other experts, and he's done this, talked about that. Now, here's the key one. Develop, maintain, and update retrieval database on hormesis. He's going to go out there and get data. Now, Dr. Kravosky mentioned that Ed's discovered this. Well, he didn't really discover it. This is a reawakening. Ed's coming along and clanging the cymbals and saying, hey, everybody, look, this stuff has been around. You forgot about it. And it's time to reinvestigate it, especially when the current models haven't been validated. So developing this database, where you can easily get the information, is going to be critical. And then use the database to publish new studies, which he has done, and to develop hypotheses as to, if you're going into translational medicine, you got to develop hypotheses as to how you're going to use this tool, how are you going to hit this hormetic zone. How do I know your hormetic zone is the same as mine? We have biomarkers for it. And to understand that, you got to understand species different individual differences and responses between it, and you need biomarkers so that you know you're in the hormetic zone. You need quick ones to identify. Okay, so here's another metric. So so this is number of citations on the ordinate here. And you can see these are the years, and the years, you know, in the 80s, you know, what, 20, 30 citations to mention of hormesis. So the Air Force, we started working in 1998, and you notice that we funded them each year. And I think this is correlative. It doesn't mean that it's cause and effect, but it sure is awful suspicious that as soon as Ed got into it and started going gung-ho, that all of a sudden this past year we're shooting for over 8,000 citations. Now, so that means, and it's also, the other people are using the term hormesis in their studies. You know, before it was Ed. You just go, well, what did Ed publish this year? And you look, oh, hey, he published it. And nobody else. And so somebody reads Ed's article and reads that article, and the next year they're going, oh, hey, I'm going to do an experiment on that. That sounds really neat. So they start publishing. Then all of a sudden everything goes, you know, exponential. All right, so to prove this is just this is just simply saying the guy who first observed it, it was in the 1880s. All right, and he was looking at yeast growth, and he was looking at things that would suppress the growth of yeast, in this case formic acid. And he did the study that Ed's probably going to tell you about that he did in your greenhouse 50 years ago. And that is that what happens is, as they got a low dose, he got more carbon dioxide coming out. They became more active metabolically. They weren't being suppressed. They were doing the opposite. Okay? And so, anyway, he went on from there, and he followed that up for years, except he made one mistake, and that is he got associated with Arndt, and which is, Arndt was a guy who was in homeopathy. Homeopathy is not hormesis. The two words both start with H and O, and so they probably figured, oh, they must, you know, forget, it was probably the same thing. Well, it totally did. One says that you can actually get a response if there's no chemical. You dilute the chemical down so much that there's nothing in that water, and you put that water in a biological system, you're going to stimulate it. That's not what Schultz meant, but it got conflated, and it became a problem because then it got marginalized. But between the 1880s and 1930s, there were many people that were very much interested in this, and these are, you can see, Nobel laureates and their graduates were actually doing research in it. There were many publications, especially area bacteria, fungi, and plants. But in the 1920s is wrongly equated with homeopathy, as I said. Modern medicine was taking off about this time as well. You know, antibiotics were being discovered, and you know, in the pharmacology field was getting big. Pharmaceutical companies started to grow, etc. But it became marginalized, and his associated with so discovered, flourished, forgotten. We went to the forgotten area until the 80s when Ed actually, with Lucky and Sagan, read their articles, went to their meetings, I believe, got stimulated, became hermetically stimulated, just got a little bit of information, and so he took off from there. And he's like a dog not wanting to let go of the bone. He's been gnawing on this thing for years and made a lot of, and Ed, by the way, this is all a database. This stuff, this history, if you want to read about it, Ed did it. That's how he started the whole thing off. So database examples, we can go through this quickly. So, okay, so let's look at in vivo treatment, low-dose ionizing radiation. We're looking at cancers here, okay? So the kind of thymomas, leukemias, but let's just look at all malignancies. So basically, when you get down to these low doses, you see something here that goes below control. So there are, and these animals right here have fewer tumors than the actual control animals that weren't exposed to anything. They were eating their normal chow and doing their normal thing, and they had more tumors than the low-dose ion. But if you take the curve up here, then it gets more toxic. All right, so this is sort of saying low-dose ionizing radiation could be sort of a therapeutic tool of some sort. I mean, you know. And then what about, look at the immune system. What's the immune response in these mice? So live mice were treated with similar types of low doses. And this is now, what we saw before was the J-shaped curve, right? Disease. Now we're looking at immune, right? If it gets bolstered up, so we're looking at the beta-shaped curve. And here we see, okay, whoa, we get a low dose here, nothing there, but then the low dose, the immune system is being activated in all these areas. There are different measures, different endpoints. So it stimulates the immune, then you say, well, that sort of makes sense, right? I mean, old people get cancer because their immune system, not just because of that, environmental factors, but basically the immune system gets suppressed as you get older. And so if today they have, what's the big field in cancer research is immune therapy, immunotherapy. They're finding out how to stimulate the immune system to attack the particular cancer. Well, it seems like low-dose ionizing radiation may be doing the same thing. Okay, so here's something else. Look at this. 1900 to 1940 clinical studies showed enhanced therapeutic effects of low-dose ionizing radiation against what? Against infectious diseases. So that means these people had what? Carbuncles, like boils, or pneumonia. And these are papers that were published. Where were they published? They were published in the Journal of JAMA, American Medical Association, the Journal of Radiology, New England Journal of Medicine, the creme de la creme. I mean, the whole Journal of Immunology is like the top journal. And they're showing, hey, yeah, this works. Look at this. You can treat pneumonia with that. You can treat disease, bacterial infection of the eyelids. You can put, gangrene and peritonitis. No, this is before antibiotics, right? Antibiotics, I mean, it was discovered in 1928 by Fleming, but it didn't really they couldn't scale up and produce this until World War II came along, and then they had to really, jack up their efforts. The other thing, though, I'll say one thing, is that, and I don't show the studies here, low-dose ionizing radiation, and Ed wrote a paper or two on this, actually can treat arthritis. So this is the other enigma here. So we're saying, wait, we can treat bacterial infections because we're jacking up the immune system, right? And da-da-da-da. So if you jack up the immune system, how does it treat inflammatory disease like arthritis? That's jacking down the immune system. So there's a thermostat, a biochemical thermostat in our body that we're not aware of, that a low dose of ionizing radiation is sort of reset button. Hits the reset button to sort of equilibrate everything in your body. And for example, Ed wrote another paper on pneumonia showing that a lot of times the pneumonias aren't dissolved and people have trouble is because it's inflammation. They kill the bacteria, but the inflammation is so rampant in the lung that they can't cure them. Then they give them, prednisone, these other things to try. Well, you got low-dose ionization does both at the same time. Gives you the antibiotic and lowers the inflammation. And must work that way, right? So that's impressive. So the hospitals, 50% of the people at operations prior to 1928 were dying of gangrene, right? The bacillus infection. You come in for an operation, well, your chance 50% of getting out alive just from, got the appendicitis out, but you just died, right? Okay, you just died because you had an infection. Well, look at this. They started doing low-dose x-rays, and after they did the surgery, and from 50% up to the 40%, all the way down to about 5%. Okay, so this sort of fits in with the last slide, right? And this is actually used. It was practical prior to antibiotics. But antibiotics came in, and a marginalization of hormesis, and we went over to antibiotics. So everybody's using antibiotics. So think a little bit ahead. If you've got a bug in you and it's sort of adapting to an antibiotic, I mean, maybe you could just, if you got low-dose ionizing radiation, that would help the antibiotic as well. Could be sort of a joint treatment. If they believe low-dose ionizing radiation didn't cause cancer at every little beta particle emitted. So this is I read these two studies, and Ed document. Actually, this one is by Myron Polycog, who's a good close friend who deceased several years ago. Wrote this a great study in dose response, and it cites the two Harvard studies where non-Hodgkin's lymphomas, you got people that were going to die because they were refractory to the normal therapy, chemotherapy, okay? Just didn't work. So that's it. Well, they got enough of these people that they said, let's give them low-dose ionizing radiation. So they gave one group low-dose ionizing radiation cohort, and the other cohort came in that just had non-Hodgkin's lymphoma, and they gave them the typical chemotherapeutic treatment. And they followed them for 10 years. At the end of 10 years, 50% of those that got the chemotherapy, the standard treatment, were still alive. That's good, right? It's good they got treated. 84% of the people who were refractory to the chemotherapy that were going to die 10 years later are now still alive. Two studies done by Harvard The Japanese picked it up, Chinese are picking it up now. No more studies in the United States because of radiophobia and because one beta particle on the linear, no threshold thing is bad for you. So when Ed if he gets up and starts talking about Herman Muller, he's the guy who set that in motion. So that's why he's so doggedly trying to find out how did they validate the LNT. All right, so database, the other thing Ed wanted to do is show, there's a lot in ionizing radiation field on hormesis, but in the chemical field, it's a totally different story. People thought maybe one chemical every 5,000 or something would do this. So Ed had a database, he had to go, well, in order to do this, his retrieval database, he had to get an entry criteria set up, because not every study you can use. You need the statistics have to be done right. You got to have enough samples. You got to have all the stringency. So you have an entry criteria and a evaluative criteria. If they pass these two, it went into one pot, and this would be the pot in which we could analyze the papers to look to see if we had a hormetic response. So that's what this retrieval system did. And when he pulled it together, these are some examples. Look at, here's cadmium, a metal. All right, what's this? This is the beta curve, right? Same thing, we don't have to go. J-shaped curve. They're measuring tumor incidence here in the J-shaped curve. Cell viability. More cells survive, fewer tumors on the animal. If we look at Agent Orange, dioxin, we see the same thing. And my point here is that this was the bottom point. There were points up here, and they drew a straight line. They said, oh, LNT, we go right through 0 because we know. And so this is the standard. So when they're protecting you against Agent Orange, they're actually protecting you against a range of doses that could be good for you. I mean, maybe it's not so good to stimulate. You know, people who are brought up in an immune, sterile environment, they have asthma and all these other, you gotta be exposed to educate your immune system. There are places where you can, well, this is the same concept, basically. This is not rocket science, really. It's really basic common sense, except nobody's unifying all these terms and concepts, and we've forgotten about them, and everybody's off doing their own little thing. Oh, and here is just to show you, there's the first dose response. This is with DDT, but if you look at the lower dose range, guess what? Another J-shaped curve in terms of cancer. All right, bacteria. Hey, here's penicillin. Low doses of penicillin, so what happens? The bacteria are growing. This is a growth thing, so this is the beta shape. So low doses of penicillin actually increase the bacteria. Yeah, so we got a couple things here. You got the genetic issue where you're naturally selecting. If you're taking your dose of antibiotics, you go too far. I mean, you still got some bacteria there that are going to then genetically mutate and develop a resistance. Well, if you're also at that low level, this is saying intragenerational, because developing natural selection, that's extra generational, that's transgenerational. You got to have the things replicate. But before they replicate, they're also stimulated to grow. So it may be a double whammy. You don't want to get too low because you're going to cause them to grow faster, and then that means they even have a better chance of natural selecting and developing the resistance. And this is showing you in hospitals when they give all these, you go in and wash your hands, et cetera, et cetera. Well, who's worried about the dose? I mean, you just go in there. But if you're in this dose, if you're putting that on your hands, bacteria seeing this dose level, you're actually encouraging their growth. So these things, these dispensers, aseptics in the hospital may actually be not good. And they may be good, but you got to know where this range is in order to use the information to benefit society. But you won't do that if you're ignorant of it and don't care or work against it. And this is ethanol. We all know ethanol, right? A couple drinks a night, and your HDL level goes up, and your LDL goes down, and you have fewer heart attacks. And then here is also, it says, in birth weight. Usually a higher birth rate is pretty good, right? But you got to hit the hormetic range. You don't want 4 drinks. You want 1 drink maybe, depending on your weight, depending on your metabolism, all of those factors. So the adaptive response, hormesis, and preconditioning. And I'm probably already over time, right? So the adaptive response, the adaptive response right here says that if you've got a cell culture and you get a toxic substance, and you treat this cell culture with a hermetic dose range. That means below the toxic level, no overt toxicity. Just treat it, and then walk away, and then come back several hours later and get a killing dose of that, and give them the killing dose. And it's a killing dose because it's been shown previously to kill, okay? And you got to control it. Well, guess what happens? They don't die. They don't die. So you go, whoa, this is the adaptive response, 1984. They studied this, and biologists have no problem with that. And so Ed's going, wait, adaptive response? That looks a lot like hormesis, right? And so he went back, and just a paper came out this year, he took all, hormetic doses and showed the adaptive dose is the same as the hermetic dose, and if you intervene in a pathway and cut off the adaptive response, you also cut off the hermetic response. So they're linked mechanistically, they're the same thing. And preconditioning, the medical term, all those doctors read, is the same thing. You precondition with ischemic reperfusion or hypoxia, and you can actually prevent severe damage to the heart, the brain, if you come with a traumatic event. So this is the adaptive response. And this is it in pictorial form. So here's the 100% thing. And so when you give the hermetic dose, right, if you hit this dose range right here, there's the doses down there, low doses, hit the doses, you're going to get some adaptive response in the thing. So if this is growth, you're going to see it manifested in growth. Okay, so you do that, you give the low dose, and you get that. But sometimes you won't see it, because, if I take a low dose of something, maybe it's juicing up my antioxidant level, and now my DNA repair is going higher, but you don't see it, right? But if I gave myself a low dose, and then give myself a high dose, a killing dose of ionizing radiation, I don't die, you're going to see that, right? Because I'm still here in front of you. So this is what this is the protective response. And it's just exact, Ed shows, it's the exact shape as the other one. It falls in the exact same range. Ergo, hormesis equals adaptive response equals conditioning. He's unifying the terminology in the field right now. This is going to take a little while to set in because, people, and scientists are very traditional, conservative, in spite of the fact that I thought when I got in the field, scientists should be broad-minded and open and better. But once they get into a track, they get siloed sometimes. And here, a high school student, which Ed actually knows all about, he invited him to our conference, and he won an Air Force prize because he showed rotifers were treated with first a low dose of copper salt, didn't do anything to them, and this is the graph after the high dose treatment. They were given an LC50, an LD50, going to kill 50% of the rotifers, okay, with the high dose, except for those that received the low dose in this range. And they basically survived the whole killing. So this is a high school student, went back, did that. This guy, he was he was a Rhodes Scholar, right? Went on the Rhodes Scholarship, and Ed's communicating with him, trying to get him to collaborate with him already. That's how Ed works, constantly doing that stuff. All right, so he's done accomplishments, retrieval database. It is real. It's generalizable across species, endpoint systems. It is not rare, and he's shown in some critical papers at least 50%, and that was conservative analysis done by him and his team, that at least 50% of all chemicals are going to show this. It's not 1 in 1,000. This should be the default system. And then it's quantitatively, it characterized the quantitative aspects, it unified the terminology, and 500 mechanisms, specific, he's documented. And now he's also, he's already shown it, the fall of hormesis was really because the lack of strong support is conflation with homeopathy and a rise in modern medicine did all that. But now he's working on the LMT model, and he's looking, and he's finding, and if you want to read, there's one paper in Edkins, one he did recently that actually summarizes all this stuff. And it says, if you'll read that, he says it was based on flawed studies, ideological bias, misrepresentations of the data, self-interest because they could gain grant funding money, dodgy review because they really didn't even review it. This is National Academy of Science in 1954 when there was radiophobia going around. They're supposed to sit down and make a decision. They were being rushed to judgment, and they didn't have time to talk about it. So it was not and you can go to the records, it's not validated. They never published this stuff. They never published a follow-up to the public, which they do now for all things. Just this, because this is where it's going now, and I want to non-ionizing radiation. Okay, everybody's afraid of ionizing radiation. What if non-ionizing radiation can do the same things? Okay, that is, if we give people exposed to radio frequency, your cell phone, or low-level light, doesn't even have to be laser light, or electromagnetic fields, such like a low dose of electricity. Can they do the same thing? I'm going to skip that slide. So, in tune with that, he switched the theme of his conference over to translational medicine to see how not only are we going to go forward to these other new modalities, but we're also going to try to talk about how do we translate it into applications. And the new modalities will get us around the radiophobia that I've been harping on. So we had last week, Ed set up a workshop on transcranial electrical stimulation. And this is, you put an anode and a cathode, and you put 1 milliamp, you won't even feel it. This is not shock electricity. And this is 10 minutes before you go into a math class. And then you do this 10 minutes before you go into math class, and then you compare the people who didn't get this pre-treatment. And the people, Oxford study, they learn faster and learn longer and retain, I guess retained is also retain more information. So that's what that workshop is about. Also can be used for neuropsychiatric treatments. Now Columbia is looking at it in terms of doing a paranoia, schizophrenia. Low-level light. Well, actually, the football teams use this to recover quickly from injuries and contusions, etc. But they don't know anything about how it works. So the guy from Harvard and a few other people have been studying this, and it enhances wounds healing, relieves pain, mostly anti-inflammatory, sort of like low-dose ionizing radiation. This does the same thing. You got to hit the right light level, and you got to hit the right light dose. That's why we need research in this area. You just can't put a battery to a flashlight to your head and say, okay, it's going to work. You got to know what you're doing with that. It relieves muscle fatigue, increases recovery from stroke and TBI in animals. And last thing here are the evidence of tissue interactions with weak and not, well, this is the cell phone. And I just want to show you, this is NIH showing you that non-heat cell phone study where on the left, cell phone is on, you can see what's lighting up. Your brain lights up and responds to the radiation on your cell phone. And it's not the heat, they've eliminated the heat. This was a great study. All that shows is that it's increasing blood flow, basically, through it. But if it increases blood flow, it's doing it somehow. It's working on a nitric oxide mechanism or whatever. Okay, this adaptive response, this is the last technical slide. Look, this is a cell phone. This is cell phone radiation. And let's look at just day one. And this is control cells in a petri dish. No radiation, no cell phone pre-treatment. This is mice. I'm sorry, this is in mice. This was given to whole mice. They were exposed to the cell phone radiation. No, there was no cell phone radiation. And we're looking at DNA damage, comet assay length. So the bigger the number, the more damage there is. So then you pre-treat. Here's an RF. This is the pre-treatment, 900, just low dose, very low dose, but pre-treated. And this is, they come back with the high dose, right? Remember I talked about high? So here, 3 gray, that's pretty darn high. And they get with the, they get a lot of damage, okay? And so this is the radio frequency pre-treatment plus the high dose, okay? And look at, in day one, if you do it right after you get the exposure to the cell phone radiation, you don't see much difference between the 12 and the 11. So it's not doing much. You get a 25% decrease in damage. But look as you go out to day 3, day 4. Let's look at day 5. If you do this 5 days in a row on your cell phone, and you come down, you still got the same control, no radio frequency. If you do radio frequency, it's the same number, right? Low dose, so it's not harming you, radio frequency. The high gray, that's the control, the positive control, still 12, right? That's solid. And then the RF, the radio frequency pre-treatment, this is the adaptive response, plus the high gray, and you're down to 4. That's a 91% protection. Tell me that the radio frequency isn't doing something to the biochemistry of the cells. Now this is hormesis, right? So we're hitting a hormetic zone here. But if hormesis is true, it says you can also go off into the toxic range as you go too high. And there's data out there to show that can happen as well. So it's very controversial. Interphone, the phone companies are studying that. But the point is taken. What if you knew how to control this? You could treat, you could use therapy on people using, you won't have ionizing radiation issue. And you're affecting the epigenetics of it. That's what you're doing. So Ed, okay. Hormesis revival, the awakening. The hard work, persistent talents, and unstinted dedication of Dr. Calabrese and his collaborators played a significant role in developing the concept of hormesis, in planting its seed and in heightening its awareness and legitimacy across international scientific community. Numerous publications, citations, scientific presentation, conferences, unrelenting workshops, debates, collaborations, inclusions of the hormetic non-linear dose response in today's toxicology textbooks. He's got 5 more offers to write 5 more articles and 5 more textbooks. All of this is the revival, the reawakening. And now he's moving his vision, the vision of the new paradigm, low-dose NIR, or photoelectromagnetic biomodulation research, offers the possibility of using hormetic concepts, if we can show that they're constant and know how to control them, to yield inexpensive, they're cheap, they're non-invasive, they're short, they're painless, they're quick acting within a day or so, they're long-lasting vis-a-vis the 6-month cognitive enhancement study, that unlike drugs, you can target and localize to specific tissues. You take a drug for a neurologic problem, you got to take it through your whole body. Localize specific tissues with virtually no collateral damage and few to no side effects. This is a grand vision, but it's all the foundations of all this. Is requires people not being afraid of hormesis, understanding hormesis and knowing how to apply it. And Ed's the leader in the field. That's why he's the champion of hormesis. There you go.
Thanks very much, Walt, for your presentation. It's a bit overwhelming for me anyways. I'd like to thank basically the biology department and Mary for making this thing happen, and I'd like to thank essentially all the biology faculty at Bridgewater of the past generation. And we had about 9 or 10, and really all were excellent. I'm going to obviously focus on Dr. Howe today, but everyone was actually pretty darn good. So in any case, I'd like to start off kind of on my personal adventure, kind of how did I get here? How did it actually happen? I'm actually a townie. I'm an original Bridgewater townie. I was born in Brockton in a hospital and moved essentially a couple days later to Spring Street, which is right off the way. And essentially my folks were essentially first generation. Their folks were true immigrants. My father's side, grandfather came from Italy in 1896. And my grandmother in 1900, they somehow lived in the same town in Italy and met by accident in New York City. Fell in love, got married, and life happened. And essentially my grandfather dies of the flu, and my father's one of 11 kids, and ultimately makes it to the 8th grade and leaves and goes to work. My mother, same kind of story, she's Polish, and her mother dies when she's 2. Her father becomes an alcoholic, and he becomes pretty much useless to the family. She and her 5 or 6 siblings get raised by an uncle, a divorced uncle, who's not even related to the family, who becomes really the father substitute. My mother wasn't so lucky as my father. She only made it to the 6th grade. And so basically my folks, unlike many other people of that generation, they really weren't alone. But they had, you might say, their challenges as they were going forward. And that's really the home that I was born into and raised. My mother and father were, my father from Brockton was a shoe factory worker, but all around Spring Street were shoe factories, if you go down there and you see where they were. And so, but as it turns out, I have some interesting connections to Bridgewater State, before it became the university, but Bridgewater State College then. My mother actually was a cleaning lady in Bridgewater State. She had a little apartment, a little dorm room in Tillinghast Hall. And I didn't actually know that until I was walking with my dad across campus, and he points up to the second floor at a corner in Tillinghast and says, he said, when I was courting your mom, that's where she lived. She was a cleaning lady, and they gave her a room up there. And actually, when they got married in 1938, that's when she left that room and they moved into Springfield and Spring Street, Bridgewater, and stayed there, and that's where they both died out of. And they were married for 62 years, and he lasted 5 more years than she did. And so, and actually, I got kind of lucky when my father quit being a shoe worker and became a janitor in the Bridgewater High School, because at that point, I think he actually started to appreciate education and saw educated people every single day. And while he had many stories to tell about some of the teachers, he really picked up the idea of the value of an education. And convinced me that what I should become is a teacher and a coach. And so I was going to become a teacher and a coach, and ultimately go to Bridgewater State Teachers College, because that's what it was called when I was a kid. Now, along the way, I have an older brother, 4 years older than me, and he was a smart one in the family. And he basically went to Bridgewater, and great things were expected of him. He was one of the kids who would make these rockets with his friends back with Sputnik and so forth in the day. And so as it turns out, he lived at home. And one day, my mother was washing his clothes, and in his pants pockets, she pulls out some papers, and she finds, this is a freshman year, first semester, and she finds 5 notes that indicate the halfway mark that he's flunking 5 courses at Bridgewater. All 5 courses, his current grade was an F, including biology with Dr. Howe, by the way. It's not botany. so as it turns out, I was there at the time the little conversation happened. It wasn't loud or anything, but my brother, he was going to do better. And so by the end of the semester, he had a 0.7 or 0.8 on a 4-point scale. It wasn't too good. And so, but he got a few up from the Fs to the Ds, but not too many. And so he was really going to crack down the second semester, and he got a 1.9, came out to about a 1.3 or 4 average, and at Bridgewater at the time, it was a 1.5 you needed to graduate. So my brother flunked out of Bridgewater State College, to the great disappointment of my folks. Ultimately joined the Army, and ultimately got some maturity, and ultimately went back to college, graduated, became a PhD, and actually was a professor at Ohio State University. So actually the rigor and the imposition and making him hold to standards was good for him. So the best thing that happened to him was him flunking out. And the best thing that happened to me was him flunking out. And the reason for that is that I was pretty much an average student in high school. Out of 200 kids in my class, I think when they gave the rankings, I was about 86th. I wasn't really in the top. I wasn't like National Honor Society. It wasn't any of that sort of thing. Wasn't terribly inspired. But when I was going to go to Bridgewater, I was, as they say, scared like crazy. Because I was going to have to, if he flunked out of Bridgewater, there was absolutely no hope for me. Absolutely none. I knew I was actually dead. And I didn't want to take biology because I'd have this person, Dr. Wall and Dr. Howe. And I figured that if I'm dead. And so what happens, I actually became a student for the first time in my life. I mean, it was because I was afraid of flunking out, but at the end of the first semester, and back in those days, my brother told me this story, and I heard it. You'd sit in the big auditorium over in Boyden, and the dean of academics came in, and they said, look on either side of you, because one of you 3 will not be here when that season starts the next year. And my brother told me that story, and I heard the exact same words 4 years later. And I said, well, I don't want to be that missing person. And so I actually became a student, if only for fear, but in the course of it, I actually started to get inspired and to like things. And I was in an auditorium kind of like this where I took biology. And I actually found inspiration and great enjoyment from it. And after one semester, I did very well, and I realized that, wow, this was fun, and I could do it. And the next thing I knew, I was a history major. I transferred into biology, and another interesting and important thing happened, and we had a very tough course called invertebrate zoology in the first semester of my sophomore year, taught by Dr. Wall, and it was kind of a weeding out sort of a course. And we went in with about 50 to 70 or so bio majors and came out with 24. And it was a very tough course. I might have got the only A in the course, but it wasn't significant getting the A or the only A. What was significant for me ultimately plays into my story is that Dr. Wall, he was an insect guy, but he got a nice grant from the U.S. Public Health Service to study the next generation of pesticides to try to replace the organochlorines and looking at organophosphates, and was stimulated by a book by Rachel Carson that was called Silent Spring that was published in 1962 that all of you should read. And well, as it turns out, I went to work one summer of '67 down the Cape. And really, as a result of the inspiration of working on environmental matters, I would eventually change my field to go to try to become an environmental toxicologist. And it was really because of that initial inspiration. So as it turns out, the real story here is that I become a junior, and I have Dr. Ken Howe as the professor in plant physiology, and he is just giving us a ton of work to do. We had to do 36 or so experiments in the lab, write each one up like a paper, and just going actually kind of crazy. He comes into the lecture room in November of 1966, and he says to the class, he said, this study on dose response where you're treating peppermint plants with this synthetic growth inhibitor called phosphon is actually stimulating the plants. And it's supposed to be inhibiting these plants. It's not supposed to happen. And he raises the question that he doesn't know why. And he'd like to find out why. It's a deviation from his experience. And so he encouraged any of the students in the class to come back and see him and to see if they can figure out what was going on. Well, as it turns out, semesters back in those days, went into the mid-Januaries, and so I actually went to see him soon after the semester was over, being the only one of the 24 kids who wanted to do this. And as it turns out, he got me to start all over again, the experiment the way it was originally done, and then to try to figure out what the class did that might have been different than what was there. And from the best that we could figure it out, it looked as though our group that was making the stock solution made a 10-fold dilutional error, and ultimately gave our plants the 10-fold less than probably what they should have. And if we kind of did it the way he said, they would have got more. And so he wanted me to do an experiment that would then have a broader dose range, the high doses that he was requiring, the lower ones that we ended up giving. And when we did, we got this, like Walt's curves, a low-dose stimulation and a high-dose inhibition. And as it turns out, this is my original notebook. And if you go into it, you can actually see that the first entry was February 9th, 1967. Mint cuttings started. February 16th, mint cuttings potted. February 27th, mint cuttings treated. And then it just kind of is filled with all kinds of experiments, data, and the like. It ends up having 2 notebooks kind of full and getting a little bit old. And then also I was able to dig out the old notebook of all the work that Dr. Howe made us do. I got an A- on that assignment. And everything was written on, my old Olympia typewriter. And it's all laid out. And so anybody who's interested to see what we were doing 50 years ago, in the good old days in plant physiology, can kind of see it. And as it turns out, in 1968, Dr. Howe somehow got me to write an abstract and somehow sent it to the Eastern Colleges Science Conference. and got my abstract accepted, and it was to be held at Yale University. And so I drove down to Yale, and the furthest I had driven at that point, I guess I wasn't really, I was a real townie. I hadn't traveled like people today. I probably, you know, by my junior year, I had traveled as far as Boston. I went to a Red Sox game. That was pretty much about it. And so, hadn't had a plane ride, nothing. So so this is going to be my longest trip. I was going to drive down to New Haven, right? so I did, had my little abstract here. And so I was so excited, this young kid who actually hadn't gone even to a different state. And so I got there early, and I find the room, and I go in there, and I'm just sitting in the back. I'm, you know, basically a little shy. And I see this, I think it was a Yale undergrad, and he was talking to this girl, and he was going to be the moderator for that session. There are many sessions going on at the same time. And maybe, I'm not sure what the interaction was between them, but he says, and this stands out to this day, he says, look at this paper. It's, I mean, the effects of phosphon on peppermint, really, what a joke. And I'm in the back of the room, really? And so I didn't say anything. I just kind of, I took it, and then ultimately calls everybody up once the room gets crowded, and he goes, and he says, well, what are you doing? What are you doing? And he says, and what are you doing? He says, I'm the guy who's doing the phosphon and peppermint that you commented on. And so he just looked at me and didn't say anything, and life went on. But it was one of these things that it was the beginning of, I should have known that that's what Hormesis was going to be like for my life. It was just prophetic. And then just to let you know, actually Dr. Howe and I published a paper on that work. It's the stimulation of growth of peppermint by phosphon, a growth retardant. I never knew the term hormesis. We just called it a low-dose stimulation and a high-dose inhibition. And actually, I graduated, and one day I was walking outside his office, and you know how professors, departments especially, they put up little flyers, you know, the assistantship open at this school or that school. And it was May of '71. And I applied for an assistantship at UMass at Amherst in physiology and toxicology. It was kind of late in the season for a lot of reasons. I suddenly became able to leave a teaching job I had to go there. And so I essentially went out to try to get a PhD in really toxicology. And so I eventually did, went out to the University of Illinois, and after 2 years there, came back to University of Massachusetts, but this time in the School of Public Health. And fast forward to I guess I'd call it 1985, and I have never heard the term hormesis. Life is going well for me in many other ways, and because how do I get bitten twice by this dose-response bite? Because this was like peppermint and dose responses. That was the earlier life of me. I got a new life. It's 20 years later. I mean, my folks were still alive and still in Bridgewater, come back and visit, but I was talking about new things. And then I got a flyer came by my office, a brochure, and I read it, and it talked about this thing called radiation hormesis, a conference out in California in the end of August of '85. So I read it, and it was a biphasic dose response. A low dose of radiation might reduce cancer, high doses might make it worse. And I said, biology is biology. Biphasic dose responses must have something in common. I don't know what it is, but they must have something in common. So it must have been a quiet day, but I called this guy up who was in charge of the conference, and I told him the story I more or less just shared with yourselves about peppermint and biphasic doses and so forth. And so he invites me out to the conference to make a presentation. And as it turns out, I never made it because, I got a graduate student to go because my wife was pregnant with our second kid, and the baby was due too close to the conference day, and I promised I just couldn't go. So I didn't go. Student makes the presentation. She comes back, tells me about the conference, and I didn't get excited at all. It was just like a paper on a resume, and that was really it. And so I can tell you that in my life I have a few professional goals, and a number I haven't really been successful at. And my one goal that was really big for me was to get an article published in the journal Science. And so by that point in my life, I was 0 for 7. And right now I'm 0 for 40. And so I'm still trying though, and may get it someday if I get. I think I get the idea now. I'll be 0 for 41 pretty soon. But as it turns out, this guy writes this article in the journal Science. It was a debate on Hormesis point-counterpoint. And when I saw this article, it reactivated my interest again. It was like getting a booster shot. And I called him up, and it was the conference director, and whom I didn't meet, and that because I missed the conference. And as it turns out, I was really disingenuous. I've been so envious that I wanted my own publication in Science. And so I called him. His name was Leonard Sagan. I said, Leonard, congratulations on your paper in Science. It's wonderful. It's all great. And then I went on to tell him that he missed a great opportunity. He just rehashed what was in the conference proceedings, and how many times you get these opportunities and you blew the opportunity. and he was so psychologically secure. He didn't get defensive, and it was really a great lesson for me. He just kind of took it. And then he said, well, Ed, what should we do? And I said, this is my opening. Academics are money-grubbing people. We need money. We search after all funding. And so I said, and I knew that he was a medical director of the Electric Power Research Institute. I knew he had access to money. So I said, Leonard, I need $20,000 to run a little get-together at UMass. And you sponsor it. I'll organize it. And he said, I'll get you the check in 2 weeks. Literally. So I got a $20,000 check in 2 weeks, and in May of 1990, I brought together a group to rethink strategic planning on Hormesis. And it was from that bite that my whole next 6 years went forth. And I'd have to say that my work was up and down, and I didn't do this because I was a great believer in Hormesis. I had seen it on the peppermint. But I was more or less looking at it as a new funding line. I had 3 areas that I was being funded in, but you always want to invest in something new in case something goes down. So it was something I was going to devote only 3% of my time to. And so I got this in, I'm fumbling around, life's okay. It's not really taking off. I actually tried to give the project away to 2 or 3 people, and they just wouldn't even take it. And so it was like this fiddler crab sticking to my finger. I couldn't get rid of it. But I kept moving it ahead. And then late 1990s, I got lucky, really lucky. I got the Texas Institute for Chemical Technology. They called, and they essentially wanted to give us a grant. Also, the Nuclear Regulatory Commission did as well. And the third a third will be Walters, and then also the Exxon Mobil Foundation. But the Texas one is interesting, and it's how you relate to money and what you will do. So they're based out of Texas A&M. So they wanted me to go down to Texas A&M to meet their board. So I went down there, and they wanted me to take a look at show Hormesis in humans, to choose a couple of examples, go into it in great depth. And I told them that I would never do it that way. I said, if Hormesis is real, it has to be evolutionarily based. You have to see this in plants, you have to see it in microbes, you have to see it in vertebrates, you have to see the different levels of biological organization at the cell, at the organ, the organism level. I said it should be, I have many of these qualities. I said, that's its strength. Its strength is then its generalizability. And they said, it's our money. And I said, it's my time. And they said, it's our money. And they refused to fund me. I said, well, let's just have lunch and talk about football or something. And that's literally what happened. So I flew back home, forgot about them. They called me back in a month, and they said, you win, we'll do it your way. And so but I had to fly down there again. And so I did, and came back with the check. And then one of these guys, Walt mentioned him, Myron Polycove, a good thinker, was with the Nuclear Regulatory Commission. Said, you're doing this on chemicals. I want you to do the same with ionizing radiation. So I kind of doubled the amount of money I had coming in very quickly. And then Walt came in to help out. And the next thing I knew, we had some resources. And then you can begin to see the upswing of all those publication citations in Walt's grant. And so but the big thing happened for me was I had about 2 initial publications and kind of lower level journals they got in. I just wanted to test the waters. Then I sent 6 papers in a row to Talk Sciences, a top journal in my field, and I got all 6 papers rejected. Decent reviews and rejected, 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 all rejections. And at my stage of the game, I mean, I still get rejections, but I shouldn't get 6 in a row. And so I called up Walt and said, Walt, I'm really working hard, but I got 6 rejections in a row. And Walt says, those are the journals we want you to get in, Ed. You have to get into those journals. And he said, how are you going to do it? What's your plan? Like the good old academic that I are, I told Walt I needed more money. And he said, what for? And so I said, I want to have a meeting, and I want to debate these guys. That editor-in-chief who was just rejecting me, I want to debate him in public. And then the guy who made his name on low-dose linearity, I want to debate him in public, and everybody else who made their name. And I'll have a big party at UMass, and I'll pull a Fidel Castro. I'll talk all one day on Hormesis, let them come in and beat me up the next day. Then we'll see where we're standing. And actually, in January of 2000, that day happened. And so I went in and gave the best ever and only 6-hour lecture on Hormesis that anybody could ever tolerate. And then the second day, I literally, was taken out to the woodshed pretty much by the biostatistician who pointed out serious flaws in what we had, and I had to admit in front of the whole audience that he was right, that I had serious design flaws in my database. It was like a very painful PhD defense in front of a much bigger audience. And then the editor-in-chief of the journal Talk Sciences, he says, I don't care what the biostatistician says. In toxicology today, we worship at the altar of mechanism, and until you have good mechanistic understanding, you're not going to get into my journals. So I basically got my rear end kicked in public. But all these guys, they were helpful. It actually was, none of it was personal. It was actually very professional. And I was so, just sucking it up, what they were saying, and kind of came back and said, well, if I'm going to get, all these people are gatekeepers. They're actually gatekeepers. They are keeping you out. And you have to get past these gatekeepers if you're going to be significant in this world. And they are telling me, that's it was great. Tell me what I needed to do to get better, actually. So I actually, the biostatistician, I had to create a whole new database, worked a whole extra, I didn't ask Walt for that money, I wasn't that crazy. And I had to work extra to make that happen. And we came up a year later with the first ever frequency of hormesis in the literature. And it was very significant. And then it took me a longer time to get this mechanism thing, but 2013, Walt alluded to it, we published a paper with 400 different mechanisms down to the receptor and cell signaling stage for hormesis. I think it's pretty much a very good, excellent thing. Now, but politically, it's very interesting. For reasons, when the frequency paper was going to be published, I thought this was my science paper. So I sent it to Science, and they sent it back in 48 hours saying, no, we're not going to even send it out. So I said, well, the heck with them, I'll send it to Nature. So I sent it to Nature, and this is actually unbelievable. Nature sent back a rejection within 8 hours. They weren't going to send it out. It was very quick. I've never seen that before. But the interesting thing is I sent it to Talk Sciences, and that paper actually did get into Talk Sci then. But within 2 weeks, I got a letter email letter from the editor-in-chief of Nature inviting me to write an article for Nature. And as it turns out, I write to her and I says, her name is Maxine Clark. Dear Maxine, I says, 2 weeks ago I sent you the very best ever paper I ever did on hormesis, and you rejected it within 8 hours. I mean, really? And now you want me to write this thing? And so she writes back and she said, sometimes the left hand doesn't know what the right hand is doing, so just write the damn paper. That's literally what she said. So I was very happy to write the paper. And so when it came out on the exact same day that the Talk Sciences did on the frequency. It's kind of an amazing coincidence. But they put us in the publicity package. And the publicity package meant everything, because that meant everybody thought you were something at that point. And once that came out, I got interviewed by just about every big newspaper. Got my picture, a big story in U.S. News and World Report. The Wall Street Journal did a whole big column on it. The Boston Globe ran a front page story. My dad was still alive at the time. that made the poor old guy, 93 years old at the time, really happy. And actually, the Journal of Science, I can't get a paper in, actually did a 4-page story on hormesis and stuck my picture in there. My mother-in-law was going to assisted living at the time. I told her, my picture's going to be in Science, it's going to be great. So she said, good, I'll show it to all my new friends. So it came out, the picture made me look like I was 250 years old and suffering from some scurvy or something. And so she saw the picture and she said, I'm not showing that to my friends. And so they never did get to see it. But in any case, I got a lot of publicity. Hormesis became a lot more known. I became much more well-known. And a lot of invitations and other activities followed. I got a lot of followers that were positive, and I got at least an equal number that were negative. And some of them, I began to be seen in really diverse ways. It was became much more controversial, got into lots of debates. I was invited since that time, probably 15, almost 20 times to Europe, into conferences to speak, debate, and so forth. Called over there to, and this is how they do it there. The Society of European Society of Toxicology invited me to debate people on hormesis. So I show up, 400, 500 people are in an audience. I debate somebody, each is up for an hour and a half, and then they vote to see who wins the debate. Yeah, it was actually interesting. Then my challenges to Herman Muller that Walt alluded to, I got invited back to, this time to the UK, to the Society of Environmental Mutagenesis over there to debate on this. That was a very hostile crowd, I can tell you that, because I was challenging the fathers of the field, and it was a bit tense at times. And so, but nonetheless, you kind of go and learn and listen and confront it all. I've had lots of debates in many different settings. And as Walt alluded to, there are many people who support what I do and think that I'm like the Joan of Arc of hormesis, so to speak. But there are others who think that I'm essentially the devil incarnate, and I am eroding their life's work, because they believe that if you take my thing to the logical conclusion, that environmental regulations will have to be mitigated to incorporate hormesis within the standard setting framework. And that has resulted in people writing books, pretty much attacking what I do, or writing articles that are pretty inflammatory and attacking. It's also, to some extent, it's flattering, even though they say, you have no character and you're a schmuck and you have no ethics and you're destroying the world. And recently, actually this year, recently there was a 35-page article written about me and my MO. And I had never seen this. I haven't shown you that one, Walt. I mean, I actually read that without my blood pressure going up. So I'm getting even able to handle this better. But the world that I live in is turbulent. It's actually very turbulent, and it's also very exciting because the science, I believe, is on my side, and basically, and it's interesting. We didn't have time to really go into it all today, but so many different fields in the pharmaceutical world are really based upon hormetic dose responses, whether they're anti-anxiety drugs, or they're anti-seizure drugs, or they're osteoporosis drugs, or many other types of drugs. The whole principle and the preclinical data on animals is all based on inverted U-shaped dose responses. This whole area of preconditioning is really all hormetic, and it's really designed to protect you against strokes, heart attacks, enhance the functioning of stem cells, enhance skin grafting, lots of other things. It's a very exciting world that we're in, and actually, for me, it all started at Bridgewater. And it all started in a plant physiology class. And it all started because the professor saw something that I didn't see, and maybe nobody else saw. He saw something that was unusual, and he wanted that answer. and whatever it was, he threw it out there, and I basically responded to it. And my life hasn't been the same since, actually. And so I have to say that my life was profoundly changed by my experience at Bridgewater. It was profoundly changed by the observation and encouragement of Dr. Howe. And I have to say he was a pretty tough mentor. He was a very tough mentor. He was unrelenting in making sure that I, in trying to learn the process of science and replication and doing it right and designing and not really taking, well, it's okay if you didn't do it right, trying hard wasn't the answer. You actually kind of had to do it right. And he was very tough. And so, toughness in many ways is, even though I don't like it when you're going through, is like with my brother flunking out, it came back to be a good thing. So I basically, one of my sad moments, amongst other things, I always loved to come back to Bridgewater and visit. And when my folks were alive, which my father lived up to 2006, I'd come back, every couple of weeks for a visit. Outside by the greenhouse, you probably will see it, there's a little lecture thing, you can stand up and talk. When my little kids were like, 4 and 5 years old, when names are Davy and Brian, I'd say, well, why don't you get up and give a little lecture on something? And me and the little kid would sit up on the little stairs. David might talk about, who knows what that age will talk. And we'd stand up and clap when they go. Then I asked the other one to go and do it. And so I always liked these little family visits back to Bridgewater. And I would walk through whatever and hit the science building and see if there's a faculty member who still is around and wants to talk and the like. So Bridgewater is pretty much deep into my roots. Even today, I went back to the old Burnell School, which is the Harrington thing. That's where I went to school there. And I hadn't been in that building for, it's been 6th grade. And I wanted to see how they ruined my elementary school and made it this business school building, that sort of thing. But in any case, as you can tell, I love Bridgewater State. I love being a townie. And it made actually all the difference for me. And so I thank, I'm just a thankful person because for me, I still have the same enthusiasm at my age that I did the day I walked in to see Dr. Howe. And thank you very much for showing up.
Guys, we do have a lunch prepared for, and really anyone who wants to join us is welcome. So, but I think a couple of questions. Do you guys have time for a couple of questions?
I can stay here till midnight. You want to talk Hormesis till 3? But any question you want on about anything.
Have you found a dose response where the response at low doses is actually worse than the high dose?
Well, it's interesting that you raised that. Yeah, people in the radiation world have there are arguments, for example, and there's an effect called the bystander effect. And the bystander effect can be both positive and negative. And so you might hit with I'll call it a single particle, could hit a cell, and that cell gets hit, and it sends out chemical messages. And sometimes those chemical messages have been observed and reported to be harmful. And so it could magnify the harmfulness at a low dose. Other of these bystander effects can be very protective. And the area of bystander biology is active. Now, very, very, I don't want to call it contentious. It's just exploratory, but there's something going on down there. And I've seen it on the hormetic front where you could give like 1 molecule per 3,000 cells, and you could increase the overall biological activity by 40% of the overall group. And so there's some very interesting biologies going on, the knowledge of which we really do have to better understand to help us all, both to avoid harm and to gain benefit, and to know when to use one and when to avoid the other. So this is a very exciting time. So there's a great search for it. It's a great question. Sure.
So I assume people have done experiments for example, growing cells in culture, hitting them with low-dose radiation, looking at changes in gene expression over the course of days, and seeing things come up and then in turn protect them from the higher doses. Have those been done, and how many examples have actually mapped out, say, changes in gene expression that correspond with the protection?
Well, I'd say probably many hundreds of those experiments have been done. Typically, on the mechanism side, sometimes they do look at gene expression. A lot of times they're looking at intervening to try to understand a receptor. They're also looking at a cell signaling pathway, and then they're trying to see if they can essentially identify which pathways are mediating this effect, and then are there receptor or pathway communications. Sometimes those studies will also include specific gene activation components. Though the gene activation components, from what I can see, for the most part, at the present time, they're not very clarifying because of the complexity of so many genes that are ultimately affected. The clarifying aspects to me so far is when they all tie into one or two pathways, and then you can figure out the pathways. In a handful of cases, people have not only identified receptors, pathways, but they've actually come down to switches. And so they actually almost have a complete mechanism. They haven't necessarily understood the genetic relationship to it. So there's a lot of dynamics that's going on, but a lot of work is on the gene activation and suppression side too.
Under the umbrella of homeopathy, are there actually medicines that are put out there, homeopathic medicines that aren't zero dilution, but they actually do have low-level, potentially, hormesis?
Yes, in a broad look at this, I spent a lot of time dealing with homeopathy and with hormesis. And I have to tell you that I have been invited over to Europe on 2 occasions to go to the 2 biggest homeopathic companies in the world, Waron and Heal, and have given hormesis presentations, discussed, debate, and did things with them. And I've had a couple of entire issues of journals devoted to trying to clarify the relationship between hormesis and homeopathy, and dug out the history of this. In the world of homeopathy, it's very opportunistic. In theory, they deal with a high dilutionist thing, but they also have many medications that are measurable, and would fall into a traditional framework. And so I find homeopathy it's easy to disparage and to characterize, but like is true with most people, we're complicated. And we have many different features to ourselves, and they do. And I think that there are certain aspects of homeopathy, especially Walt alluded to conditioning, in which there's the possibility that certain homeopathic treatments, I believe, could work in a post-conditioning sense and be examples of hormesis, but it needs to be studied. I've written about that, published on it, and it's interesting. When I decided to do that, people told me, don't do this, because you'll discredit yourself by writing in this area and making this claim. And the way I look at it is you put it out there and you have to defend yourself. And if I'm going to write it up, usually I hope 100% of the time, I'll be able to explain and defend and document what I'm saying, even if I'm going out there and saying that I believe that homeopathy could exist within this post-conditioning framework, and that could be hormesis, and I'll stand behind the data that I have that said it. So those are going on, those debates, and I'm kind of always in your face sort of a person.
All right, guys. So I think we'll meet upstairs. If anyone wants to join us for lunch, please feel free if you have more questions to ask. But I want to thank you very much, Dr. Gazzoco, Dr.